Look to your left. Now look to your right. By the time the three of you were 5 years old, one of you would have contracted human cytomegalovirus. In fact, over half of adults over the age of 40 have the virus. How come a virus so widespread isn't that well known, you ask? That's because the people infected with HCMV show no symptoms. It is a virus that remains in the latent state following primary infection. The virus can be reactivated under certain conditions, the most common of those being immunosuppression. This makes it a danger to any immunocompromised individuals, meaning those with HIV and AIDS and any other immunosuppressive disease are at risk, as well as those that have received organ transplants, as they are on medications to suppress their immune system so they don't reject the organ. The virus is also a major risk to newborns, as it is the most common cause of congenital birth defects due to viral infection. Almost 1 out of every 200 babies is born with a congenital HCMV infection. And with around 4 million babies being born a year in the United States, that's around 20,000 babies a year to have HCMV infections. Current treatments for the infection are focused on drugs that inhibit the viral DNA polymerase. However, more treatments need to be developed as resistance to these drugs is rapidly developing. In order to develop new treatments, more needs to be known about the virus, specifically everything about its replication cycle, the proteins that it encodes, and then the cellular components that these proteins interact with. So what is already known about human cytomegalovirus, or HCMV? Well, it is the largest human herpes virus with over 190 open reading frames. Other than that, it is a DNA virus. The DNA genome it has is surrounded by a protein coat called the nucleocapsid. The nucleocapsid is surrounded by a layer of proteins called tegument, and all of that is encompassed in a viral envelope. The mechanisms by which the viral assembly occurs is still not completely understood, and the function of many of the tegument proteins is yet to be determined. However, it has previously been shown that 2 of those viral tegument proteins interact with each other, forming a complex that is essential for viral replication. Those 2 proteins are UL94 and UL99. The UL stands for unique long, so it's unique long 94 and unique long 99. The relationship between these 2 proteins, when it is abolished, completely inhibits the production of virus in the infected cell. It is believed that the interaction between these 2 proteins is necessary for the completion of viral assembly and envelope formation. Specifically, the protein complex interaction is needed for proper localization of the viral assembly complex near the nucleus, and then it moves to the envelope formation site near the membrane. It is believed that a cellular protein is also involved in this process and plays a critical role. After this hypothesis was made, a complex screen of cellular proteins was performed to identify proteins in the cell that interact with the viral protein complex. Through this screen, it was found that the cellular protein Rab33b interacted strongly with the UL99-UL94 tegument protein complex. So a little bit about Rab33b. It is a protein belonging to the Rab family. Rab proteins are proteins that function as regulators of intercellular trafficking, and one of their main roles is moving cell surface proteins from inside the cell to outside the cell. Because of this, it is a logical target for viral hijacking to help with completion of viral assembly and envelope formation by moving the assembled virus to the membrane to form its envelope. For my project, I will be using CRISPR-Cas technology to generate a stable cell line not expressing the protein Rab33b, and then infect it with HCMV and then monitor viral replication. So CRISPR-Cas technology is a form of genetic editing where if you know the sequence of a region in the genome that you want to remove, you can remove it. We already have a cell line that is permissive for HCMV infection, and it is already expressing the Cas proteins required for CRISPR technology. So we will perform the knockout, and once the knockout is performed, I will confirm it by using a Western blot, which is just a tool used to measure protein expression. So once we see that Rab33b is no longer being expressed in those cells, we will be able to infect those cells with the human cytomegalovirus and then monitor the viral replication. It is expected that the virus will halt in its replication cycle just before envelope formation, and then if this link between Rab33b and the UL99-UL94 complex is shown, it can be further researched to characterize specifically how the interaction between the two occurs. And then years from now in the future, this could eventually be the target for a new antiviral drug for human cytomegalovirus, focusing on disrupting the interaction between Rab33b and the viral protein complex. Thank you.